So it's a rather grandiose title. Really what you're going to get today is a bit of a run through the garden. A lot of what, in fact, most of what I'll be talking about is actually work that's been done over the years by people at Intelligent Optical Systems. Let's see, it looks like it should be that one. Nope, that one. Which one? Okay. No.
Keyboard. Or mouse.
Oh, okay. So this is just the monitor. Okay.
Yeah.
All right. Yeah. That's the backup. There we go. It's got to be one of these buttons. No, that's
so let's
not
just
it.
pull the keyboard out.
Okay. There we go. Good enough. Oh, I got to close that. No, I don't want to end the show. Just click the screen. There we go. That's better. All right. Well, you may or may not have seen the abstract. But this is not work that we've done in our company. This is one of my favorite slides. It shows up in a lot of talks I give, so I apologize if anybody's seen it before. This is an early optical biosensor. This is actually an optical blood pressure monitor, invasive blood pressure monitor. And, you know, the guy who's taking the reading has got the easy job. There's his two lab assistants down there holding the horse steady while they puncture an artery and stick that glass tube in it. You know, they got the hard job. But this is an optical biosensor. This guy is optically reading in real time the blood pressure of this horse.
Is that considered to be minimally invasive?
It depends on if you're the horse, I guess. But this is also an invasive optical blood pressure monitor. This is something that I did actually way back when I was at Bell Labs, long time ago. There are various incarnations of this. This particular one has a polymer, a flexible polymer. We were using silicone, among other things. Has a little reflector in it. And then there's a partially reflective layer. Turns out you can just use the glass area interface. You get enough reflection there. You don't really need to put metal down. And so you form a very small Fabry-Perot cavity. So you get optical interference between the light waves that are reflected from that surface and the light waves reflected from that surface. As the blood pressure, as pressure changes, it squeezes this down. And you get a shift in the spectrum. You can read that out by capturing the spectrum, or you can just look at like one edge of the interference curve and look at the intensity go up and down. And at the time we were working on it, we were told by physicians that they really, they do a lot of invasive blood pressure monitoring using electrical probes. This thing was attractive because it can really fit on the end of an optical fiber. And back in the day, they were always hair-thin optical fibers, but fibers are very small. Anyway, since that time, a lot of people have done work on optical sensors. I really meant this one to be at the end, or should be at the end, since we talked about the internship thing. So I'll maybe come back to the introduction or the overview of the company a little bit later. But what I really want to focus on here is this aspect of the work that we do at IOS, Intelligent Optical Systems. So this is something that we were just joking in Bruce's office, something we used to call the submicron chemical sensor. Obviously, in these times, you can't say submicron, so this is nanoscale probe. What you're looking at here, end on, and I think I might have a better picture sideways. There you go. What you're looking at here is essentially a near-field scanning optical microscope tip. So that tapered fiber goes down to, and I should point out there are 3 separate optical channels in here. If I back up, you can look end on. There are really 6 different points. It's that one, and that one have actually got optical cores guiding light. But of course, that's a trick because, this is smaller than a wavelength of the light we're guiding. So it's really a near-field optical technique. in the end, what happens, and I'm not giving away any secrets because we actually got some patents filed on this. In the end, what happens is the light gets to the very end and squirts out the tip using surface plasmon resonance as a means of transmitting the light. So it's actually plasmon. I could say plasmonic nanosensors. Really, if I wanted to use two buzzwords in one sentence, I could say that. But what we do is we take this tip, we shine light into one of the optical channels, dip it into a monomer solution that has a UV curable polymer in it, and an optical indicator dye. So in the end, what you wind up with is a little bead, a little dot, exactly in the near optical field of light that's squirting out of that little tiny sub-wavelength hole. And here's even a better picture of it. So this, in this case, I think this was probably a pH indicator. But you shine light down here, and then you look at the fluorescence. Basically, most of the time we use fluorescence. You look at the fluorescence of this thing, and it can tell you what the local pH is in an intracellular measurement. And what we were able to add to this is work originally started at the University of Michigan, my alma mater, by Raul Kopelman. But what we were able to add to this in collaboration with Kopelman's group is the ability to do multiple channels all down in a sub-wavelength space. So this is some work that we are actually poised to continue up at Intelligent Optical Systems, possibly with some collaborations here. We'll see. So going up a little bit, but not that much in length scale, we'll now look at a fairly important application. Just as a bit of history, an awful lot of money was spent by an awful lot of companies on developing an intravascular blood gas monitor. The so-called blood gases, pH, CO2 partial pressure, and oxygen partial pressure are a fundamental measurement that's done in emergency care and critical care. Basically, your pH and PCO2 and PO2 start to go wild about 5 or 10 minutes before you go into deep shock. So it's really important to be able to monitor these things in real time and in critical care units. Nowadays, I mean, even today, you still have to take a sample, run it to the lab, and get the measurement. So an awful lot of work has been spent on trying to make this a real-time measurement. What we have found in collaborations with some people at the Los Angeles Biomedical Research Institute, LA Biomed, up at Harbor UCLA Hospital, is that basically in the little space between your eyelid and your eye, the partial pressure of oxygen, partial pressure of CO2, and pH are very similar to what's found in the bloodstream. And in the case of a premature infant or a neonate, it's really hard to take enough blood to do a blood gas measurement, let alone do that 5 times a day to make sure this kid is not going into shock. Furthermore, there's a really important reason to measure oxygen near the eye because most of these kids are in incubators, and there's a very fine line between giving them enough oxygen so that they survive and giving them so much oxygen that they go blind. So it's really important to do online measurement. We found that yes, they blink a lot, but you don't have to stick them with needles. But we found that by putting this probe in here, you can actually wind up making a very good real-time online measurement. You want to make it as small as you can. So what we've done in this case, instead of putting it on the tip of an optical fiber, we have actually drilled holes in the sides of optical fibers and embedded the pH, PCO2, and PO2 indicators in the fibers themselves. So you could theoretically do all 3 measurements on a single fiber. The probe that we've actually packaged and are hoping to do some clinical trials on in the next year or 2 is 3 separate fibers. We get some fairly good readings of O2 concentration. CO2, we've got very well developed optical sensor technology. So moving right along to yet another subject. This is obviously a DoD-funded project U.S. Department of Defense funded project, this particular one. But our company has, over the years, developed a great deal of expertise in multi-target lateral flow assay technology. This particular technique is one that's familiar to a lot of people. It's seen as in the standard pregnancy test. An awful lot of lateral flow strips are used in glucose testing, for example. So the format itself is one that's very familiar, very cost effective. What we've been able to bring to this is the ability to quite easily measure multiple analytes on a single test strip. So for those of you not familiar with it, the way it works is you put a sample down on one end, and I guess you could call this picofluidics. It's actually just capillary action that moves the sample along this strip. And as the sample moves, it picks up various reagents and encounters various other substances. I'm going to keep this at a relatively simple level. So you can label with antibodies and fluorophores your targets. You can do essentially a sandwich immunoassay, all without any moving parts, all just on this miraculous piece of, well, it's not paper anymore, but it's essentially like really fancy filter paper. So now this is an example of basically, at one point we were looking at biomarkers for cardiac events. This is an example of a myoglobin assay at different concentrations. Here's an example of how you can do multiple assays on a single strip. So there are actually four different readout lines here. And this, if you can see the picture, you can see two very bright readout lines. This is a plot of intensity. You can use different colored labels to also get multiplexing on your test strip. So as well as physical location, you can use color. We project that you can get about a dozen assays on a single test strip if you work it right. We typically use quantum dot labels just because they're so convenient. Here's a very pretty picture of multiple color quantum dots in multiple locations on a test strip in a little reader that we've made. Note that in this case, it's not just the simple looking for the plus or minus sign on your pregnancy test. You're actually using a much more powerful readout technique to get a quantitative measure. So it's not just the fact that you have fluorescence on this capture line saying, yes, I have something there that I'm looking for. By putting it in the reader, you can quantitatively determine how much of your target is there. And we've worked on a number of different assays. One of our most active projects, and the reason why the first slide showed a guy in a combat helmet, one of our most active projects is with biomarkers for traumatic brain injury. And obviously this is a major problem for military personnel and also people in various contact sports. But traumatic brain injury is now called the signature injury of the current conflict because critical care medicine in the field of combat has gotten so good that people who would in the past have died from loss of blood or other massive injuries are now surviving pretty serious blast injuries. And they're finding that these people, while physiologically they seem okay, it turns out that the shock wave and the blast effects that cause the major trauma also cause hidden damage within the skull. So traumatic brain injury is a very important thing to be monitoring. Mild traumatic brain injury, otherwise called a serious concussion, can have lasting effects that people were sort of unaware of. And the ability to measure biomarkers, proteins and peptides and other biochemicals that are released into the bloodstream as a result of this kinds of trauma. The ability to measure that is very important. We've also found that these same biomarkers can be used to look at the process of treatment and recovery. So in addition to triage, where you say, okay, this person's got a serious brain injury, they need to have their skull trepaned so that you can relieve pressure, that's one important application. But another very important application is, okay, this person walked away, he's fine, but now it's 2 days later and these traumatic brain injury biomarkers are getting more and more concentrated in the bloodstream. You really have to do something. Or conversely, this guy, was 50 yards away from a hand grenade that went off. He's telling everybody he's got headaches. He's malingering. Put him back on the front line. So the military is interested in both sides of the coin there. But I don't want to go into too much detail on the biochemistry behind it. I just want to point out that this particular platform is one that is very, very convenient for not just field medicine in military applications, but also for third world health considerations. It's a very, very cheap, very, very durable format. I mean, these test strips can stand high temperatures. They're easy to store. You just keep them in a sealed bottle and put your sample on them. Quite easy to operate. And the reader that we designed is one that is such that you really just have to stick the test strip in and it gives you a number out. So, and of course it uses a very small amount of sample. And you don't have to use blood, although the particular traumatic brain injury one that we've been working on up to now has been using just a single drop of blood. It turns out that biomarkers for a number of interesting things are found in just about anything you can sample. So we've looked at lacrimal samples, we've looked at saliva, obviously urine can be sampled for a number of things. And we've, as it says, we've looked at traumatic brain injury, breast cancer, cardiac events. Leishmaniasis is a parasitic disease, which is very common in sub-Saharan Africa and also in India. Interestingly enough, it's of interest to the military as well, although most of our funding on that project has come from NIH. It's interesting to the military as well because as people go out in the field, they wind up in places where Americans don't normally go, and diseases which nobody ever heard of wind up being a serious consideration. So we've been working with the PATH Foundation. I don't know if you're familiar with that. It's an organization devoted to third world health in generating some assays for things like leishmaniasis. I should point out some of the biochemical work that we've done in the company. We do make, and in fact you can go on our website and buy quantum dots from us if you like. We do make and sell quantum dots. But we use quantum dots in a lot of our biochemical sensor work. So oftentimes we're using them as markers in antibody-antigen reactions. Excuse me. We do work with classic fluorophores still from time to time because sometimes they're just the best thing to put in there. But quantum dots are really so convenient because you can excite them with one wavelength and you can get all these different colors. And it's just we really think that in terms of labels, if you can use quantum dots, it's really preferable. Okay, I'll talk a little bit about some more of our sensor work. This is moving away from the chemical sensing. This is actually a probe that we fondly refer to as the prostate meat thermometer. It's designed to measure microwave diathermy or temperature profiles during microwave diathermy. So I think I might have a little bit better picture of the probe. Maybe not. Okay. I think in the end, at the end of these, oh, here we go. This is good enough. If you can see that, this is actually okay. That's a standard catheter that is used for urinary tract catheterization, and this is our temperature probe. This little section, as you can see, is about 1.5 centimeters long, and within that 1.5 centimeters, we're measuring temperature in 10 different places with 1/10 of a degree accuracy. So the idea is to place this temperature monitor right in the active zone where you're doing the diathermy treatment to make sure that you're only cooking the parts you want to cook and that the parts that you don't want to cook are at a reasonably low temperature. And the beauty of this, of course, is, it is an optical fiber. You can make these temperature measurements all in a single fiber in all these different places. To make 10 different measurements with thermistors or an electrical technique, you'd have to have 20 wires. Okay? You don't even want one wire in a high-intensity microwave field, particularly not when it's located in an intimate place. You all saw what happens when you put metal in a microwave oven. So fiber optics really is about the only way to do a good temperature measurement in diathermy, and the ability to make multiple temperature measurements all in a line, all in a single fiber is one that really helps quite a bit. The way it works is we use what are called fiber optic Bragg gratings. I won't go into the optical theory behind that, although I'd love to later on. Anybody want to ask me? But essentially what these wind up being is temperature to wavelength transducers. So they reflect light at a very, very narrow band of wavelengths. You can see here, I think you have all 10 of them on the screen right here. They reflect light at a very, very, very narrow band of wavelengths. And the exact and precise position of the absorbance, well, this is reflection, of the reflection peak, depends very critically on the local state of strain, how much you stretch the fiber, as well as any temperature-induced changes of the refractive index. So when the fiber heats up, it stretches a little bit, the refractive index changes a little bit, and you can very quickly measure temperature. And as I said, we can measure temperature to within a 1/10 of a degree. Here we go. Here's a little plot of the readout from one of the temperature sensors, and I think I might have a profile, and I guess not. But we were actually able in a tissue phantom to profile temperature quite nicely, and like I said, it's a minimally invasive technique. Other physical measurements related to biomedicine include electric field and magnetic field measurements. This is I guess we were calling it an OEG or an OKG, optical encephalogram or optical cardiogram. Essentially, it's a probe, an optical probe for measuring electric field, not voltage, electric field, which is really what you want to measure when you're looking at human electrical activity. The probe is the E-field probe is really quite simple. It's just like well, not the hands, but the little LCD display in my watch. You wind up with an optical polarization rotation depending on the local electric field. That's why your numerals turn from dark to light on your watch. Basically, the polarization state is being switched by an electric field. Polarization state of your liquid crystals is being switched by an electric field. Turns out that you can use that effect quite nicely to build an optical fiber probe that can respond very reproducibly and very accurately to the local electric field. And the nice thing is, since it's an electric field monitor, you don't have to gel this to somebody's chest. You can just lay it on there and you're measuring electric field. Here's a case where we actually were measuring. Let's see which one. Yeah, okay. This is an oscilloscope showing a trace of a cardiac electric field simulator. You can buy machines that are used to calibrate standard EKGs, and we were using our optical fiber probe to look at the signal, and we very, very specifically recreated the cardiac pattern. So that's another whole set of work that we do. We've also worked on magnetic field sensor as well for similar applications. Okay, back to some work that we've done using direct spectrochemical detection. In this case, we're not using markers, we're not using labels, we're just looking at the native fluorescence of, in this case, biofilms. This was originally funded by the NIH's National Institute of Dentistry. I think they're NIDCR now, craniofacial. Anyway, but it's the Dentistry Institute at NIH. It was really very interesting. The application that they were looking for was to tell whether or not the dental water supply lines had developed biological contamination. There's some inside baseball on why we got the funding when we got it. It turns out it was a fortuitously timed proposal. But in any case, I'm here to tell you when the dentist, after you wash out your mouth, tells you spit. They're supposed to clean out those dental lines, and they do. They sluice them out usually with a chlorine solution. But there is an opportunity for biofilms to grow in there, and if the wrong stuff is growing in there, you can wind up, if it sits in the lines overnight and you're the first patient in the morning, you could wind up with some serious levels of toxins in that little Dixie cup. So the idea was make a probe that you can use to snake down those water lines and just tell, okay, as I pull it back, do I see, is there biofilm there, or is, if there is biofilm there, is it active, is it not? So what we really wound up doing was making what I call a life detector. So this thing will tell you whether or not you've got live biofilms, and what we're looking at is basically the inherent fluorescence of the NADH, NADPH reaction. If it's going on, then you got something biological there. And if it's not, then you're okay. So this does detect live biofilms, and bacterial activity doesn't necessarily have to be a film. You can also detect live organisms in solution. It's a relatively simple-minded approach, but it's one that worked quite well for us. We put it in a number of different formats. I think the earlier one shows, there's this big thing right here was basically to look at biofilms on surfaces. At one point, very interesting, McDonald's and Burger King and Kentucky Fried Chicken were not interested because they don't have biofilms on their working surfaces. And if they did, anyway, but at one point we thought the food service industry was going to be a good customer for this. Turns out that in addition to, by the way, the dentists aren't using this after all. They're just, they improved their cleaning methodologies, and so I still say spit. But it turns out that the air conditioning industry was more interested than anybody else because those big cooling units, you probably got one on top of this building somewhere, those big cooling units develop bacterial mats, and they have to be cleaned out about once a year. Right now, pretty much everybody cleans them on schedule. They just go up there and they use a biocide, and then they very carefully remove everything, and away you go. It's part of what the heating, ventilation, and air conditioning people do in a big building. But that's done probably about twice as often as it really has to be done. So the idea would be we had some serious negotiations with a major air conditioning company about developing a probe that they could use to go up and say, okay, do I really have to do this cleaning now, or can I wait? So it's interesting how research applications wind up straying from these little tiny dental supply tubes to these great big honking air conditioning units. Oh, interestingly enough, the second most interested group of people were the folks that make kidney dialysis machines. On the water side, not the blood side, they wind up with a biofilm problem as well. So we're still in negotiations with those folks. We've looked at that for water quality monitoring. It's really more suitable for biofilms. Depending on the locality, you want to detect one colony forming unit per cubic meter. No, not really. It's not really that bad, but the sensitivity is not quite good enough for actual drinking water quality monitoring. It really was developed for biofilm applications. I mean, we could jack up the sensitivity, but then that would price it out of competition with just straight culturing methods. Moving on to actually this has been the subject of another one of our spin-off companies, a company called Maven Technologies, and I think if you go online and Google Maven Technologies, you'll see what they've been able to do with this. We licensed this out to Maven. I was involved for a while in the spin-off, and now it's really, they're off and running on their own. This is a real-time biochip reader. We think it's really direct competition for the BioCore, surface plasmon resonance-based biochip reader, only better, of course. The way this works is really quite simple. What we're doing is measuring the plane of polarization of light that's reflected off of the surface of a biochip. So it works on total internal reflection. We're shining polarized light in from the bottom, looking at polarized light coming out. And what we're actually able to do is in real time look at binding events. Anything that binds on the surface of this chip changes the local optical density. So actually, there's a layer of stuff that becomes thicker and also has a different refractive index than what you start with. So the way it works is you lay down your recognition elements, and we've done both nucleic acid as well as protein-based recognition elements on the surface. Lay down your recognition elements and set your optics up so that you have minimum light being reflected from the surface. You work basically right near the critical angle of total internal reflection. Don't have to be at the critical angle, but that gives you the highest sensitivity. And unlike surface plasmon resonance, the angle that you work at, as I said, isn't all that critical, because what you're looking at is the rotation of the plane of polarization, not annulling, like you are with SPR. So as you get more and more binding, you get a larger and larger shift in the polarization of the reflected light. And well, this is an early lab bench setup. We've gone a little bit beyond that, but that setup was one that allowed us to take this data. And here we're just using a BSA, a very common thing for people to put down to test their biochips. And we have a micro spotter that put down spots of various concentrations. And down here we were looking at 4 femtograms per square millimeter, which one of our guys calculated was like less than 1 molecular layer. So get pretty good sensitivity with this. We have now, Maven Technologies is going to be marketing a benchtop unit, and I think they're aiming at first for high throughput screening applications, which is where biocores are used. So pharma companies are interested in this. We've gone on and developed a much smaller version, and what you're looking at here is, this is the microfluidics system. This is the actual optical system. Let's see, this is it's an 832 screw, so the whole box is about that big, like that. That's the optics block. It still works on the same principle. And the microfluidics, and this is very important, the microfluidics part is also the same size. Lots of times you'll see these beautiful pictures in Science Magazine or Nature Photonics where you've got this really cute little microfluidic chip and all this tiny lab on a chip stuff going on. What you don't see is there's a relay rack over here with a bunch of pumps and tubes feeding the microfluidics. This is all packaged in ready to go. Put them together, it's about the size of a shoebox. So we are thinking that Maven Technologies may want to come back to us for the next generation of technology. And this is an active project within the company right now. But speaking of surface plasmon resonance, plasmonics is yet another buzzword of this century. We wind up finding ourselves doing localized surface plasmon resonance or plasmonic enhancement of antibody response. So this is technology that we have actually now gone beyond in-house. Instead of fabricating these little nano triangles or nano pyramids, they really are using a technique developed originally at the University of Illinois. We're now using nanolithography to make more customized shapes. But the idea here is to use localized surface plasmon resonance to enhance native signals. You can enhance both fluorescence and, of course, Raman using this. So the idea here, obviously, is to make the highest sensitivity assays you can with the smallest amount of material. Okay, moving on yet again to some, this one didn't come out very well, unfortunately. I think I might have some better pictures. Yeah, here we go. The company IOS is involved in, as you can tell, physical sensors, chemical sensors, and biological-based sensors, but also we're involved in the processing of sensor data. A certain category of our customer base thinks that every sensor is a focal plane array. It's just a CCD detector. That's a sensor to a lot of people, particularly in the military, but not exclusively. So again, this is more of our sensor work. In this case, what we're doing is processing data from image sensors. And what you're looking at here are images. These are photomicrographs. So the sensor, if you will, is just the CCD array in a camera. These are photomicrographs of blood vessels on chicken chorioallantoic membranes. So if you take off very carefully take off a square of the shell of a fertile egg and don't pierce that little membrane that's just under the shell, it has blood vessels growing in it. It turns out, I mean, that's interesting to know in itself, but it turns out that this is actually the model system that's used by most pharmaceutical companies when they're investigating potential anti-angiogenesis drugs. Angiogenesis is the process by which blood vessels are formed. Cancerous tissue grows really fast, so it needs a lot of blood vessels. So a major breakthrough of, about 20 years ago was the finding that these anti-angiogenic factors wind up being pretty good chemotherapy drugs. And so the search for better and better and more specific angiogenesis controlling drugs is still very active. Most of the time, the way companies were quantifying the angiogenesis that they were looking at and the effect of their drugs on angiogenesis was by taking pictures and then hiring people to measure the widths of the blood vessels, count the number of blood vessels, count the number of branch points, and then write it down or enter it into a computer. So we completely automated that process, and in fact, Igor Chernovsky, who was the scientist who developed this, added one extra thing, and he said, well, I mean, look at this pattern. Clearly, this is a fractal pattern. So we added a fractal dimension there, and we were easily able to come up with a quantitative measure, indicator of whether there's a lot of angiogenesis or a little bit. I mean, it's obvious to you looking at this, but there's a very good quantitative measure of telling how much more blood vessel formation there was in this sample than in this sample. So we've applied this to everything. I'm sorry. We applied this to everything from these types of images to CAT scan images to MRI images. It turns out that lots of blood vessels being formed in a short period of time around a suspicious mass is highly indicative of it being malignant. So image analysis is very important actually in cancer diagnosis. So this is another example of some of the work that we've done. Continuing on with image analysis, and I think I'm getting towards the end of the talk, so don't get too nervous here. We'll have a few minutes for questions. Continuing on with image analysis, we're going back to direct spectroscopy in a way. What we developed here was a little handheld probe, and this actually got quite far. We did some FDA-compliant clinical trials, and this is still a potential candidate for commercialization. We've been actively talking with some groups quite recently on this. What you see here is a little handheld probe. It's just a point-and-click spectroscopic probe, optical fibers, going back to a commercial. We made our own, but then we went and bought a commercial fiber optic spectrometer. But all the magic is in the processing of the data. And we used a combination of direct linear algebra as well as neural network techniques to characterize the difference between the spectra. And now we're looking at both visible and near-infrared spectra. I'm sorry, the wavelength base didn't come up, but we're essentially going from almost UV all the way up to 1,300 nanometers. So what we're looking at is that, I mean, it's a complicated spectrum. It's a mess. There are lots and lots and lots of papers on tissue spectroscopy and even a lot of papers on spectroscopy of burnt tissue. But we were able to find that there is a fairly easy way of telling the difference between what are called superficial, partial thickness, and full thickness burns, or used to be called first degree, second degree, and third degree burns. Very important to be able to do this within the first day or so, because what you want to do is remove tissue that's never going to get regenerated. Full thickness burns, I mean, that's dead tissue, or it's going to be dead very soon. You want to remove that as soon as possible so that you don't get infection. You want to leave the superficial first degree burn tissue because that will regenerate. You want to leave as much of that as possible, take away as much of that as possible. So what you really want to do is find the margin, the edge between where the stuff is that has to be debrided, scraped off, and where the stuff is that you want to leave. And so this little point-and-click probe was one way of doing it. Obviously, you can apply the same algorithms pixel by pixel to a hyperspectral image. And we think that that's really the way to go. Another example of what we've done at the company. Now I'm moving on. As I said, most of what we do is sensing and monitoring. And I'll back up to those first few slides and do the company overview at the end. But we do have some projects that I lovingly call our nonsense projects because they don't have anything to do with sensing. but I guess that's not really true in this case. In this case, what we have here is an adjunct for existing microsurgical catheters. It turns out that there's a real problem in microsurgery as the surgeon approaches the field where they want to operate. A lot of times they don't have a good sense of scale. They don't know how big this stuff is, and they don't know how far away they are from the surface. And the last thing you want to do is to crash your microforceps into healthy tissue. So, I mean, all sorts of techniques have been used, including placing down a little tiny scale, which, a little 1 millimeter long thing with tick marks on it so that you can see how close you are. What Vladimir Rubtsov, who's another one of our principal investigators, has put together is a system that basically allows you to get two different views, allows you to get a triangulation on your sample without having to use two separate endoscopes or two separate optical channels. It's a single optical channel, but by using a trick, a little tilting plate of birefringent material, As this thing, just ticks back and forth, it's a MEMS-operated device. As this thing ticks back and forth, it shifts the optical view left, right, and you wind up being able to triangulate. And in fact, you can use the same technique to make a 2D endoscope into a 3D. Basically, you can get stereo pair without having to add a second optical channel down your endoscope. This is a little bit too complicated a slide to brief, but essentially this shows you the results of looking at the two different angles, and what it's supposed to prove is that you can measure, quite easily know that you're 3 millimeters away from your working surface by looking at those images. And this actually has been incorporated into the working channel of an endoscope now. Other work that we've had going on with, it's another university in the California system, up in that big city up north of here. UCLA actually has a very good MEMS program. They have an NSF MEMS center there that I think they've actually cut the cord. I think they don't have NSF money anymore. But what was I saying? Oh, I'm sorry. Yes, the folks up at UCLA have developed this cute little pneumatically actuated micro gripper. These are you can see it right here. These are plates of silicon with little tiny balloons in between each one of these phalanges, or on each one of these phalanges in between each of these knuckles. And so when you pressurize them, they grip. And I don't know if I have a video of this in here. Probably not. I might. Okay. Let's see if this works. Come on now. We might not get the video. I should have brought my computer in. Anyway, what you wind up with is, as you actuate these things, this thing actually closes into a little fist and grabs whatever's in there. The problem that some of the MDs that were working with our collaborators up at UCLA found was that they couldn't see what they were grabbing. So what we were actually able to do was to incorporate right in the palm of the hand a little light source and a little imaging fiber, multi-core optical fiber, so that you can see exactly what you're grabbing and exactly where you are as the grabbing operation takes place. So we actually have 2 active programs with the National Institutes of Health, National Center for, I think it's NIBID. No, it is NCR, National Center for Research Resources, is funding this work. I really wish I could show you these videos, but I guess we're just going to have to. Oh well, that's okay. All right, let me now back up to the beginning, and I'll talk a little bit about the company, and then we'll open it up to questions on any of the stuff I've been talking about. But I guess what I wanted to leave you with was an idea of the amount of work, the scope of work, and the excitement that the people that work at our company feel in the stuff that they're doing. We have found that applying optical techniques to measurement is at one point I thought we were kind of like limiting the company. I wanted to focus the company. Turns out that we're working on like a whole bunch of things anyway. So right now IOS is about 50 people, about a dozen PhDs. We have facilities in Torrance and now up in Pasadena as well. We've been in business about a dozen years, as we said earlier, and we address not just biomedical but other markets as well. We make and sell from the building a very sophisticated laser ultrasound non-destructive test system, which is used in everything from weld monitoring to measuring the thickness of steel as it comes off a rolling mill white hot. We're well known for our work in chemical sensing, in particular the development of optical fiber sensors, the whole length of which is chemically sensitive. So this right here is a a 300-meter spool of nerve gas sensitive fiber. As I said, we do make and sell quantum dots. We're into image analysis. This is something our image analysis folks have put together for a law enforcement application. It's now being sold on an OEM basis. Our business model is to remain as an applied research and development company and to sell and support, high-end, high-margin products like our laser ultrasound, and then to spin off things like the optical chip business, Optometrics that Bruce mentioned earlier, the biochip reader that I talked about as well, the Maven Technologies biochip reader, and some other things. We do some OEM work as well. We work on physical sensors and physical optics. Probably in a certain segment of the world, I think we've gotten the most web play on our light emitting diode incapacitator, or LEDI. If you, I shudder to say this, but it really is the best way to see, to get right to what I'm talking about. If you go on to Google and type in puke ray, yes, you heard right, you'll see a bunch of video on a non-lethal weapon that we developed for the Department of Homeland Security. And that's now being deployed. We're arranging manufacturing probably within the U.S., although possibly offshore as well, on the light-emitting diode incapacitator. So in our physical optics and sensors work, as I mentioned, we've done temperature, electromagnetic fields, but also acoustic emission, pressure, strain, stress, cracks. We have a very large concentration in non-destructive evaluation and the use of fiber Bragg grading technologies. Chemical sensors, I think I already mentioned a lot of it. Two formats, this little nanoprobe, and so we have the world's smallest fiber optic chemical sensor and the world's longest fiber optic chemical sensor. But we also, as I mentioned, did integrated optics for chemical sensing, and we've done an awful lot of work. In fact, probably our most active work right now, it's between the cables and fiber tip sensors. These are little petri dishes that have little tiny pieces of porous glass, and within them are embedded chemicals that are sensitive to oxygen, hydrogen. We've done them for CO2. We've done them for a number of different analytes. So fiber tip, little things that you can put on the tips of fibers or package any other way that's convenient. It doesn't have to be at the end of a fiber, but they change color in the presence of what you're trying to monitor. Then I also talked about the advanced signal and image processing stuff. We saw a couple of medical applications. This is now a product this is one of my favorites. This is really cool. We got patents. I think we've got some patents that are going to come out quite soon in this area. It's one of these things that when the guy came into my office, I said, "You got to be kidding. This isn't original. Somebody's done this before. It doesn't look like anybody had." This is really kind of cute. Essentially, it combines very simple image capture with fast signal processing. It's only within the past year or two that it's been possible to do this in a compact way. And what we got is a remote camera. In this application, it's a camera on the end of a stick, and you want to see what the camera's seeing. Well, what's the problem with that? You have to, if you're not holding the camera so that you're exactly vertical, if it's tilted, then your image on the screen is tilted. So you either got to go like that or you got to go like that. There's this cognitive dissonance between which way you know is up and which way the camera is seeing as up. Turns out that if you just take that image into a computer and add a couple of sensors, attitude sensors on the camera, in real time now, it's possible now, in real time, you can process that image so that no matter which way you turn the camera, up is always up. Seems simple. It's really remarkable what this does for you in applications where the image acquisition is separate from the image display. And we've actually had some interest on the part of people doing endoscopy in possibly putting this on endoscopes as well. So image analysis and image processing is another thing we do. And then obviously I've talked a lot about the biosensor work that we've done. One thing I didn't mention was I talked a lot about sensor biosensors where you're sensing biological or biomedical stuff. But you can also use biological materials to sense other things. And what you're looking at here is actually a series of well, these are alive. These are biological entities. Essentially, they're little colonies of bacteria, and we're monitoring their response to wastewater. So you can tell what's in the wastewater or how healthy your wastewater processing system is. Excuse me, by looking at how healthy the little bacteria growing in these cells are. And we do that by looking at oxygen respiration, and we squirt in known amounts of nutrient, and we see how they respond to nutrient. It's yet another of the things we do. I've talked today about roughly 20%, 25% of what's going on in the company, or what has gone on in the company over the past few years. So considering we only got a dozen PhDs and 50 people total, we're real busy up there in Torrance and Pasadena. We have a lot of fun. It's very interdisciplinary. We got everything from biochemists to computer scientists to physical chemists to actually some physicists who know something about optics, and some physicists who don't know anything about optics but know about other things. Pretty good electronic engineering support staff, and a really good group of administrative people who make life easy for us. We do everything from contract research for large companies, medium-sized companies. We collaborate with a lot of academic groups, and we're actually very, very enthusiastic about doing that. And the stuff Bruce and I were talking about just a little while ago sounds great to me. And you know, in many ways, the people that work at IOS are in a quasi-academic environment, because the principal investigators there wind up writing proposals, getting funding from the government to work on projects that they think up and they want to work on. The company provides the environment for people to do that. We're always looking for bright and creative people, so that's my pitch. Like I said, the business cards are here, and it's time for questions.